This lecture is going to be on the evolution of vertebrate urinary systems, and I have a separate lecture or two on the human urinary system anatomy and then on its physiology. And so some of the terms introduced here, like kidney, nephron, are far better explained with reference to the human structures in those other lectures. So nitrogen is a critical element for living things. So if you take DNA, obviously it includes these nitrogenous bases. Here, nitrogen atoms are depicted in pink. So without nitrogen, there is no DNA. Proteins are made of amino acids. Amino acids have amino groups, which have a nitrogen atom. So without this pink nitrogen atom in the amino group, you get no amino acids or proteins. And so we need nitrogen for proteins in DNA. However, if one then metabolizes this molecule and removes the amino group, a hydrogen added to this would then create ammonia, which can be toxic in high levels. And so living things certainly need to manage their nitrogen levels. Well, the simplest living things can't. And so if you were to look at how living things are classified, and there was a modification in the old classification scheme compared to the more modern one based on genetics, where flatworms have moved. So sponges, jellyfish, and corals, and the simplest worms, the acial flatworms. These branches have no way of excreting waste other than diffusion. So the simplest animals didn't really have anything resembling urinary structures, but everything above this branch point at least has something. And as one is looking for a word which would then summarize this group, I don't think it's in broad usage yet, but the word I've seen is the nephrozoans, which would be all animals above this branch point, which then have some way of processing this waste. Now, in all fairness, however, the tubules, which are often compared to urinary tubules in flatworms, primarily function in osmoregulation, or just regulating the levels of water versus ions between the body and the environments, and so a lot of diffusion of waste is simply passed through the skin, and that's one of the advantages of being so very flat. However, these animals do have what are called Proto-Nephridia, with these being structures which can force fluid from the body, but retain other elements like proteins. And so here in gray would be these ducts. Once again, these can be associated with some excretion of wastes, but they are primarily used for osmoregulation. But nevertheless, some management of waste occurs, starting in these simple animals. As we start getting to those invertebrates related to the vertebrates, the primitive chordates, we start to see some similarities with the vertebrate condition. First, there are blood vessels, and even going back to worms, the most primitive worms which have blood vessels, the nemertine worms, have excretory structures associated with them. In the primitive chordates, there are what are called foot cells or podocytes. In the human kidney, the capillaries, which are filtered to remove nitrogenous waste, have foot cells or podocytes over them, and the primitive chordates have the same. And so, oops, I'm sorry, wrong video. So here's an important part of the human nephron, which exists in invertebrates prior to the separation of the vertebrate lineage, these podocytes. So here you're going to see the the capillaries have pores known as fenestrations, and sitting over these fenestrated capillaries are these podocytes or foot cells, which are helping with the filtration. So nothing is released from the body unless it not only can pass out of the blood, but can also then pass through the slit in between these foot cells, these filtration slits. Once again, such a setup existed prior to the vertebrates. Also prior to the vertebrates is that blood could be filtered. Blood could be filtered so that waste could leave. But this didn't create urine per se, but created what's called a primary filtrate. And then the fluid which was leaving the body then can reabsorb good stuff. This then ultimately results in what's called secondary filtrate, or the urine which is released from the body. And then in vertebrates like mammals, what comes out of the blood is not urine, it's filtrate, and it must pass through this tube, and good stuff gets reabsorbed before it becomes the urine which is released from the body. So before we get to the vertebrates, there are a couple of structures and strategies for dealing with waste, which are contained in the ancestors of vertebrates. Now, nitrogenous waste must be excreted, but there are different ways that nitrogenous waste can be excreted. And so ammonia can be produced directly. And so ammonia is a nitrogen atom with 3 hydrogen atoms. And the release of this is known as monotelism. And this is what we see in most vertebrates because it's found in the fish. So fish and amphibians secrete ammonia as the nitrogenous waste. A problem is if you're going to be producing ammonia, ammonia is very toxic. And also you need a great deal of water to remove ammonia from your body. About 1/2 liter of water is required to remove a gram of nitrogen in ammonia. Obviously, if you're living in aquatic environments, this water may be readily available. And so the advantage is if you do have access to abundant supplies of water, you can easily dissolve this ammonia in your urine, but you can also release ammonia through gills and through the skin. And so if we look at these fish or these amphibians, they are producing ammonia, and not only in their kidneys, but also in their skin, their gills, and releasing it that way. As we will see when organisms start to adapt to land, this is a problem because of how much water is required. And so the amniotes, they will then, instead of releasing ammonia, primarily be releasing what's called uric acid, depicted here. So this is what is released in reptiles and birds, and the advantage is that you don't need as much water, and this may have been related to the first amniotic eggs. So if you're going to lay an egg on land and embryos are going to develop in an egg, obviously they don't have access to water, and so therefore releasing nitrogenous waste in the form of a powder instead of something that requires a great deal of water is an advantage. And as I'll return to when we talk about birds, obviously if birds are spending time on this rock, you may see these excretions here, but this is not primarily feces, this is primarily urine. But instead of urine having urea or ammonia, it has uric acid, which if you involve sodium but potassium ions, can produce a solid, which is a way of conserving water. So that white substance on the rocks, that was actually the release of nitrogenous wastes. And so reptiles and birds, the ancestral amniotes, practiced uricotelism, where they produced uric acid as their nitrogenous waste. Mammals then produced urea, which is less toxic than ammonia, but requires more water than uricotalism. And so there are different ways of releasing this nitrogenous waste. And actually, you don't have to pick just one. For example, this lungfish and then as some of the tetrapods, such as frogs or turtles, can. I'm sorry, this frog, not turtles. They can choose and release ammonia when they are in water, or release uric acid when they are on land. So there's obviously then some variations in the strategies of how to release uric acid. Now, obviously, when I end with mammals, the focus is going to be on the kidney. The kidney is a structure which is retroperitoneal, so it is not in the same body cavity as the intestines. So if you were to depict, the intestines, the stomach, they're in a body cavity known as the peritoneal cavity, lined by a peritoneum, but the kidneys are actually behind that. If you look here in, this cat dissection, the intestines are in a space, but the kidneys are then behind this peritoneal space, and the name for that is what is called retroperitoneal. Now that is true of the kidneys of mammals, but that will also then be true of fish kidneys as well. It's not in the same space as the intestines. In fact, it's behind that space. They are retroperitoneal. Now, mammalian kidneys look like this. They are compact structures, and they form in the pelvic region, and all of the waste is coming from the filtering of blood. So there's a very big renal artery which services the kidney. And then that was if we consider the kidneys of humans or other mammals, those principles are the same. It will be different with fish, as we'll see in a second. Mammals, their kidneys are made of nephrons, which look like this. So they are all encased in a glomerular capsule. So here you have capillaries in a bag. And then you have a tubule with these discrete sections. As we start looking back at the fish, you will see that not all capillaries have to occur in a bag, not all bags have to have capillaries, and that the renal tubule does not have to have all of these sections. And so when we consider the evolution of vertebrate urinary systems, we can see homologies, even going back to primitive fish and even chordate relatives of fish. But certainly we don't see all of the human structures and the structures which we find in humans evolved in stages. Okay. And so first off, let me describe what I mean by kidney, and then we'll have to go into what we mean by nephron. So kidneys, well, we think it's straightforward, these structures developing near the pelvic region. But once we look at fish, and then even when we consider vertebrate embryos, including human embryos, we'll see that, very often kidneys are coming in three parts. The part which we associate as being part of our kidneys, that's what's known as the metanephros. But the majority of the kidneys that one would say find in, a typical teleost fish or an amphibian or reptile, here in blue would be called the mesonephros. But actually, I'd like to start off with what's sometimes called the head kidney, known as the pronephros. Now, these regions don't sound like a kidney to you because you no longer possess it, but you did as an embryo. And when we look at the pronephros, we see all of the following types of nephrons. So the very bottom nephron looks a lot like the type of nephron that you would find in a human. But there are other types which are more common in the fish. So number one, notice that here, this funnel is going to take in waste that can then be excreted from the body, going through this archienephric duct, which will, then release urinary waste from the body. So notice, however, this is not associated with blood vessels. It is simply filtering the coelomic fluid. The fluid from the body cavity can then enter here, and ciliated cells can help to move it. These cells can reabsorb stuff. All right, so it's controlling what is released as waste, so you don't release good stuff. But then also these cells can secrete additional waste. Both of those functions are what we see in the tubules of mammals. And so you see this primitive type of nephron. Can it release the waste from blood? Well, it can, because look at this bunch of capillaries. They simply open up into the coelomic cavity. It's what's called an external glomerulus. So these capillaries can release waste. The waste goes into the coelomic body cavity, and the coelomic body cavity can then be filtered and the waste released. Then there are also then tubules which do receive coelomic fluid, but which are associated with capillaries as well. And so waste could come from either one of two places, and then both, from both the coelomic cavity and then also from blood vessels where waste could be then added to the contents of the tubule and then produce as a waste. Note that in this Proto-Nephros, there can also then be internal glomeruli, which are completely wrapped in a glomerular capsule, so that as filtrate comes out, then all of it is then directed towards the tubule. Once again, a Proto-Nephros could have all of these kinds. Once again, indicating that there seem to have been stages in the development of these excretory mechanisms. And so the waste ends up going into an archienephric duct. Here you can see it under the microscope, all right? And then this would then run the length of the body here in this hagfish. So you can see lateral to the dorsal aorta and the cardinal veins, you would see this archienephric duct, which is then collecting urinary waste from each segment. So the urinary structures would be segmented and then eliminate them from the body. So that's the Proto-Nephros. Who has a Proto-Nephros? Well, hagfish have functional pronephric kidneys in both the larvae and the adult. The lamprey has functional pronephric kidneys in the larvae. The jawed fishes vary. Most jawed fishes have a functional larval pronephros, and a few will have a functional adult pronephros. In amphibians, the larval pronephros can be functional, but not typically. And then a few nephrons from the pronephros might actually contribute to the adult kidney. And so this pronephros obviously is originally an early form of the vertebrate kidney that then gets gradually replaced through various lineages. By the time one gets to the reptiles and mammals, the pronephros is no longer functional. So then what is functional in these higher vertebrates? Well, if the kidneys form kind of as a line down the back of the body, the pronephros could be called the head kidney, and everything else can be called the opisthonephros, which can then be divided into the mesonephros and the metanephros, the metanephros being the one which mammals will retain. And so the opisthonephros is then those two combined. Now, in a comparative anatomy textbook, sometimes instead of a separate chapter on urinary and reproductive, you might see then some references to the urogenital system, combining the two, because these ducts, so this mesonephric duct, this can be used to, say, release sperm. And actually, this develops as part of the mesonephros, say, in mammalian males, but it's the mesonephros which will become part of the vas deferens in human males. And so are these structures urinary or reproductive? Well, they can be both. So here you can see the mesonephric kidney in the lamprey, which is releasing both urine and gametes into this archinephric duct. So kidneys in fish are not what we think of as kidneys. They stretch the length of the body, and you can see that here's the kidney. Here is the male reproductive tubule, the ductus deferens, and so there can be an association between the urinary system and the reproductive system. Now, it is retroperitoneal in all gnathostomes, and so as you just saw there in the shark, it's not in the same space with the intestines. Instead, it's behind the lining of the ventral body cavity, the peritoneum. So you have to actually pick the peritoneum away before you can actually see the kidneys clearly. And so once again, the intestines would be in this space, and the kidney here in brown, you would not see that dark brown structure until you picked away at the peritoneum. So these kidneys run the length of the body, and they are retroperitoneal. Now they are associated with the reproductive organs, and even when you look at, say, amphibians today, look here at this salamander. Here's the kidney, here's the testis, and look, there are reproductive ducts which move sperm from the testis into the kidney. Does the kidney move urine? Yes, it does, but sperm also pass through it. So once again, that term urogenital system, the urinary reproductive system, are not separate at first. Here's the testis of a frog. Here is the kidney, but notice then here are these ducts which will move sperm from the kidney, from the testis in through the kidney. So once again, there is not a complete separation between the urinary and reproductive systems in these earlier vertebrates. If you look at a pig embryo, and just that's typical of mammalian embryos, look at the kidneys. These mesonephric kidneys actually stretch up to the arm buds and run down the length of the body, just as they do in fish. And it will once again be part of the mesonephric duct, which will then be retained in males, but not females, as part of the vas deferens. And so what we think of as the kidney is actually what develops near the pelvis as the metanephros. And then this is just odd, but we can discuss this again with the reproductive system. If you think about the position of the kidneys, they're in the mid-abdomen, and the gonads are, either at the pelvic brim in females or outside the body in males. But they actually start in different places in embryos. So the metanephric kidneys start in the pelvis, and the gonads here in green start higher up, and they have to switch places. So our kidneys and gonads do not form in the sites where they will ultimately reside. The gonads have to go down and the kidneys have to go up. And sometimes there are birth defects where there can be, say, a horseshoe kidney or a pelvic kidney, a horseshoe kidney if the two join, a pelvic kidney if it stays inside the pelvis, but I can get back to that. And so the metanephros is the kidney, the primary kidney come mammals, and the other two exist as embryos, but will not then exist after embryonic development. So when we were embryos, the Mesonephros and Proto-Nephros degenerated, and then we were left with this kidney. But why don't I come back to that when we get to embryos? Let's go back to the evolution of these things. The mammalian kidney can have, say, 1 million nephrons. This is the functional unit of the kidney. A fish kidney can have far less than this. And so, fish kidneys are not as good as excreting as the mammalian kidneys. The mammalian kidney has this renal corpuscle, the capillaries in a bag, followed by a renal tubule, which has different sections: a proximal convoluted tubule, a loop of Henle, the descending loop of Henle, the ascending loop of Henle, very good for the reabsorption of water, and then finally a distal convoluted tubule. How did this, these nephrons, which once again are the functional units of the kidneys, how did they evolve? Well, we certainly see homologies. If you look at, the kidneys of frogs or fish, you can see, look, capillaries in a bag. So here are the glomerular capillaries in a glomerular capsule. That's similar to what we have. Look, here's the renal tubules. Lined by simple cuboidal epithelia. So that was, in a fish. We'll see this in a frog. Here's the renal corpuscle, followed by the renal tubule. We'll see it in a turtle. There's the renal corpuscle, followed by the renal tubule. So there's certainly similarities. So yes, these, there is homology. The mammalian or the human urinary system didn't simply appear. That being said, there are similarities, but those other ones are not identical. So when we see hagfish, we see that it has a glomerular capsule, all right, as do all of the other ones. So when I put this here, this would be like the most primitive animal which has this structure. So all of the craniates have a glomerular capsule, and they have the first part of the PCT, which we call the proximal segment 1. Lampreys and all other vertebrates have not only proximal segment 1, but proximal segment 2, and the distal segment, which forms the distal convoluted tubule, and the collecting duct that these tubes empty into. The jawed fish all have things that the jawless fish lack. Urea is a primary waste. They have fenestrated capillaries, which is being filtered to remove this. The JG cells, which are involved in the regulation of how much urine is produced, are known in all of the osteichthyans. This would include bony fish, amphibians, reptiles, and mammals, in addition to an intermediate segment. Mammals and birds will turn the intermediate segment into a loop of Henle, but the intermediate segment does exist in bony fish and amphibians, even though it doesn't form that loop of Henle, which is so good at reabsorbing water. The renal corpuscle gets a little bit smaller in the tetrapods, and then mammals and birds then convert the intermediate segment to that loop of Henle. So if you look at the renal pyramids of the kidney, you can see these long loops of Henle, which make the reabsorption of water easier, as you can see here. So mammals and birds would have that. So once again, a jawless fish may only have 30 nephrons in its kidneys. That of a mammal can have 1 million, and all of the tubules combined can be more than 120 kilometers. And so these loops of Henle can be quite large, and this then helps us live on land and reabsorb water. So nephrons exist in fish, and they have homologous structures: renal corpuscle, renal tubule, but you also then see a progression in the various parts as we go through these various groups. And I just kind of repeat that here. And the thing that's most interesting is if you look at the nephrons of a fish, you can still see then very primitive nephrons here. So here's the kidneys of a fish. But one of the interesting, yes, you see renal corpuscles, the capillaries in a bag, and you see renal tubules. But also you can see that in addition to filtering blood, there can be waste coming in from the coelomic cavity. In hagfish, these can actually even open into the pericardial cavity around the heart. So whereas in mammals, all of the filtrate is coming from the glomerular capsule around blood vessels, you can see in just a second that in fish, there are some of these nephrons, which are not only filtering blood, but filtering coelomic cavity fluid as well. So here you can see that. So where is the waste coming from? Well, some of it is coming from the capillaries here, all right, but then some of it is also ultimately coming from the coelom as well. And so this is a primitive setup of nephrons, which we can see here in the fish, and then in the frog and the turtle, you can see, there's a renal corpuscle lined by simple squamous epithelia, just as we have in humans. There are renal tubules, etc. Notice the space is a little bit bigger. Once again, one of the things that happened later is the renal corpuscle would decrease in its size. Okay, just a couple of quick things. One, when we were embryos, once again, we formed the Proto-Nephros and the Mesonephros, but they would not be functional as embryonic kidneys in we mammals, and they degenerate and are not present in adults. Although once again, males will keep some of the mesonephric duct as the part of the vas deferens. It's only the Metanephros which then becomes the adult kidney, and that has to then rise and change its position. Before I focus on mammals, just a few other things. The urinary bladder is not universally present. Jawless fish do not have urinary bladders, and in sharks, the urinary bladder is only present in females. Then as you get through the osteichthyans, the bladder is present in both males and females, and as you can see here in this frog, it is lined by a type of epithelium which can stretch. Normally, epithelia are named for the shape of the cells. They could be flat and squamous, or they could be box-like and cuboidal, but these are called transitional epithelia because the cells can stretch. They can look more box-like or cuboidal when the bladder is empty, and they can look more squamous when the bladder is full. And so the human bladder you see here is lined by transitional epithelia, but you can see the equivalent cells are located in a frog bladder as well. Now, just speaking of frogs, there's a bit more going on there. So this frog could be urinating and releasing water from its body. It could also essentially be drinking, because not only can water enter through the mouth, but there is a portion of the skin near the pelvic area on the ventral side where water can be taken up. So this frog could actually be taking water into its body as it sits in water through a patch of skin on its underside. All right, so first we can see that here. So here's this ventral pelvic patch where water can be taken up into the body. Now, urinary bladders often store urine, which can be released from the body. But in frogs, the bladder can actually store water that could then be taken back into the body later if the frog needed it. And water can actually pass from that ventral pelvic patch to the bladder. And so the bladder can not only excrete waste, but actually it could take up water, which could then be used. So frogs can release a hormone which is the homologue of ADH in humans, and this vasotocin hormone then causes water pores known as aquaporins to be produced, which can then cause water to be reabsorbed. And so water can go from the environment through the skin of the ventral pelvic patch into the bladder, and then from the bladder into the body. And so, there are different aquaporins. Some are only expressed in the ventral pelvic skin and the bladder. And so, that obviously a difference. One, before we get to mammals, just a note on birds. Birds lack a urinary bladder, and the way that they process a urinary waste is interesting, and it actually involves the digestive system. So once again, birds, like all reptiles, practice uricotelism, where uric acid is the primary waste. But when urinary waste are produced in the kidney, it then passes into the ureter. Notice that there is no bladder. And then the ureter then empties into the cloaca, which we could split into 3 regions. The ureter empties into an area known as the urodeum. Sorry, that's one too many O's there. And so the nitrogenous waste will pass here, but instead of then leaving the body, then pass through reverse peristalsis, going the opposite way that you would think, passing through another section of the cloaca known as the coprodeum, then going into the large intestine. Yes, up the large intestine to these sites known as intestinal ceca. Here, sodium and potassium ions can help complex with the uric acid to produce this white solid, and this then absorbs helps with the absorption of water. And so the water which, say, a mammal would release with its urinary waste is kept within the body. And then this white solid can then later be released from the large intestine. Yes, urinary waste leaving the large intestine, leaving the cloaca, and out the body. Sphincters tend to control this so that the large intestine is releasing either urinary waste or feces, but not both at the same time. And as here you can see on these rocks where birds have been, this is primarily urinary waste, the uric acid, which has formed that white solid here, which has been released from the large intestine. Okay, so obviously then there are homologies as we compare the urinary systems, but then there are certainly then differences as well. Before talking about the urinary systems of mammals, just one last thing that we observe in amphibians and reptiles, and even in birds. Normally, our circulatory system runs like this: artery to capillary to vein, back to heart. Artery, capillary, vein, back to heart. There are 2 places in the human body where we have artery, capillary, portal vein, capillary, vein, back to heart. So there's a vein in between 2 capillary beds. We call that a portal vein. There is a portal vein between the hypothalamus and the anterior pituitary, and the big one is the hepatic portal vein in the digestive system. So humans have these 2 portal veins. But if you were to look, say, at amphibians and reptiles, they have another one known as the renal portal vein. So there may not be a renal artery which is delivering blood to the kidney to be filtered. Instead, it's the veins coming back from the leg. So and some veins coming back from the leg must go through the kidney. In others, there may be alternate routes where some of the blood coming back from the leg is going to the kidneys, and otherwise it could bypass it. So notice here that there's the kidneys, and notice how blue it is because it's receiving the venous blood coming back from the legs. So if the legs are at the lower right, here you see the renal portal vein, which go to the kidneys. So the blood from the leg is going to the kidneys where it is filtered. And then only from the kidneys, then, this isn't a frog, by the way, do the renal veins leave the kidney to form the inferior vena cava or post cava, which then goes back to the heart. So once again, blood coming back from the legs goes to the kidneys here in brown. Only after that does it form the renal vein, which goes back to the inferior vena cava. And so as we look at blood flow. So here in this frog or in reptiles, there can be alternate paths so that the blood coming back from the leg has two choices where it could then reach, go back through a number of veins or not. So there can be renal arteries as well coming off the dorsal aorta in addition to the renal portal vein. And so this was something that we see in amphibians, reptiles, and in birds, although it is not as important in birds, but mammals do not have any trace of the renal portal system. So then speaking of mammals, the mammalian kidney is that metanephros, which has an outer cortex. This is where the renal corpuscles are. And then it has a medulla, and some of the nephrons then form that, that big loop of Henle. So here you can see in a mammalian embryo, there is a mesonephros, there is a pronephros. You can see it in mammalian embryos, you can see it in bird embryos, but this will degenerate and not contribute to the adult kidney. So in this pig embryo, the mesonephric kidney will degenerate, the metanephric kidney will be retained. All right, here it is in the pelvic area. And as I had said, they have to switch places. The kidneys down here, the gonads up here in a mammalian embryo, including humans. The gonads have to go down in females to the pelvic brim, in males even further, and leave the body, where this metanephric kidney must then go superiorly. And every now and then there's a birth defect where one of the metanephric kidneys stayed in the pelvis and is known as a pelvic kidney. There can be abnormal blood vessels reaching the kidneys. There can be a horseshoe kidney if the two fuse, and so the rising of these kidneys can then be problematic. Other than that, the urinary system in mammals is largely the same. So there are two kidneys, once again, the metanephric kidneys, two ureters, which drain the kidneys, one single bladder, and then one urethra, which then removes the urine from the body. This can be seen. I have a video here of the urinary system in the cat. You can see that the kidneys are retroperitoneal. There, the kidneys then will release urine into ureters, which goes into one single. So here you can see the kidney, here you can see a ureter. This then goes into the bladder. There's only one of those, and then one single urethra, which exits the body. One question then is, does the, I'm sorry, let me back up. So as I had mentioned, say in fish, Obviously, there is a connection between the urinary and reproductive systems, so much so that you could, use the term urogenital system, because it's hard to speak of them as a separate entities. Now, in female humans and some other mammals like primates and rodents, the urinary system is 100% separate from the reproductive system. So urinary and reproductive systems are completely separate. But remember, ancestrally that wasn't the case. They were joined, and even sperm had to go through the kidney, in the salamander and the frog, as I had said. That isn't true of all mammals. In about half of mammals, the urethra does fuse with the terminal portions of the vagina. And so the urinary and reproductive systems are not 100% separate. So that varies in some mammals. It is, and that would include humans and primates. But here in this cat, there is some fusion at the distal end where the urethra and vagina meet. So once again, the urinary system does not have to be 100% separate from the reproductive system in females, and it certainly is not in males, right? Because in males, the vas deferens, which is covered in the reproductive system, then comes and then goes around the ureter. So you can see it going around the ureter there, and then will within the prostate gland join the urethra, so that the urethra in male mammals is both a urinary structure which releases urine, but also then part of the reproductive system releasing semen. And so that's kind of a leftover, from the connections which the urinary reproductive systems once had. So once again, I have another lecture on the anatomy of the human urinary system and one on the physiology. This was a discussion of the urinary systems of vertebrates, and then clearly one can see that while there are homologies, even going back to invertebrates, clearly the features of the human urinary system have evolved over time.